Chapter 17 here, a couple of parts in chapter 17. If I can get my ear apparatus together here. All right, so when we talk about immunity in the human system is basically composed of a series of defense mechanisms. We've talked about the primary defense mechanism in anatomy and physiology 1 is the skin. We talked about some skin diseases last week, but humans and most other simple animals have a natural immunity as well. It's based upon a non-specific defense system such as phagocytosis, which we talked about not only in the beginning of this class, but we also talked about phagocytosis in Anatomy and Physiology Part 1 as well. And so both the AMP2 and AMP1 and AMP students that are in here are familiar with that concept of phagocytosis. And, of course, obviously the skin, as I've talked about before, the integumentary system, and the mucus lining in the openings of our system, the mouth, the nasal pharynx, and even the rectum and the vagina into a degree. These are openings to the external environment, all of which have a mucus lining that help to protect the system from invading microorganisms. Altogether, these defenses are referred to as innate or non-specific immunities. Much of the innate resistance seems to be inherited. Populations are exposed by generations and generations of microorganisms, and so relatively we can become resistant to certain microorganisms, or certainly we can live in perfect harmony with certain microorganisms just because of this development of a relationship over time. Characteristics such as different body temperatures make humans resistant to many animal diseases. Animals have certain defense mechanisms that make them resistant to many of the human types of diseases. So even though we use animals a lot of times in diagnostic studies to evaluate treatment responses to certain medications, recognize that the immune system, or those processes within the bodies of ourselves and other animals, are a bit different, and thus some diseases that we see presenting in humans will not present in animals, and vice versa. What's on big on the media attention right now is the bird flu. Right now, primarily affects birds, primarily located in Asian areas. However, it is a fear that this type of virus will mutate and eventually start to affect humans. Right now, the only transmission to humans have been with people who work directly with these types of animals. But there is a fear that it will mutate and begin to be able to be spread from human to human. Right now, that's not the case, but obviously it's a big concern about that. The virus initially started only affecting birds, and so only birds got sick from it and humans did not. So there's always the potential that microorganisms can mutate in such a way to be able to infect humans. Also, the other thing we have to consider, there are certain disease processes in man that can compromise our immune system enough that we can't fight off diseases, and so different microorganisms that typically would only affect animals or typically would live in our system without causing disease, because there's been a compromise in our own immune system, then now we're susceptible to those diseases. Higher animals also are protected by adaptive or acquired specific immunities, and we'll talk about that a lot more. So, review: innate defenses against any pathogen, immunity, specific antibodies in our immune system and our lymphocytes that respond to an antigen. An antigen is a foreign invader, something that's not a part of our body, considered to be an antigen, gets into our body, and our immune system responds to that, attacks and tries to get it away. Antigen, substance that causes the body to produce specific antibodies. Now, don't confuse the antibodies that our system produces with the antibodies that we take, antibiotics that we take. Physician writes a prescription, nurse practitioner writes a prescription, gives you, and you go get an antibiotic filled prescription, different from the antibodies that's produced in our body by our immune system. And then proteins, antibodies basically are proteins that are made in response to an antigen. So our body makes antibodies that will attack the antigen, the foreign invader. Types of immunities, types of acquired immunities, refer to specific defense responses developed in response to an antigen, which are organisms or substances that can provoke the production of special proteins called antibodies, certain specialized lymphocytes, in particular B cells and T cells. Naturally acquired immunities These are obtained by natural exposure to antigens, for example, a disease organism. If you get exposed to a flu virus during this year, your body will develop antibodies, and you won't get infected by that same microorganism, that same virus, again this year. Now, for viruses, because they mutate, there are a number of different viruses. It doesn't mean that you won't get sick again this year. You may get infected by another virus, but for that particular type of virus you got exposed to, you developed antibodies to it. You get exposed to chickenpox when you're small. Your body develops antibodies against chickenpox. You don't get chickenpox again for the rest of your life. So we can develop antibodies once they form, once they're in our system. Typically, we don't get reinfected by those organisms because our system now has antibodies to be able to fight off that microorganism if we're ever exposed to it again. Now, another term that you should be familiar with is called subclinical infections. Not that I tested you on this, but just the concept. Subclinical infections, you don't have any evidence of symptoms at all, but you can confer immunity. So, for example, herpes simplex virus that causes a cold sore. 99% of the population has antibodies to herpes simplex type 1 because we all get exposed to that virus. Now, we all don't get cold sores. Many of us do, but we all have the antibodies to it because we've all been exposed to it. If you get an active infection, you will have antibodies to it. Unfortunately, those who present with the symptoms, you get a blister or a cold sore. Now you have an active infection. It actually lives on a nerve, and it actually will sit dormantly on the nerve and will continually represent itself. Now, individuals, just because they have the antibodies doesn't mean that you'll always have symptoms. But once you've had the symptoms in a cold sore, which is a little different from some of the other organisms we talk about, you have antibodies to it, but unfortunately you will always present with the symptoms because of how it lies dormantly. But many people can get a subclinical infection, never present with symptoms, but you have developed antibodies that will protect you, again, against those invading microorganisms. Naturally acquired passive immunity, you'll need to know all these definitions. Naturally acquired passive immunity, it basically involves the transfer of antibodies formed by the mother to her infant. This may be done by a transplacental transfer, so while the baby is still in the oven, you'll get a transfer of antibodies by the mother to the child through the placenta, the blood supply from the mom to the baby. And then it renders the infant immune to most of the diseases which the mother has already been immune to. Now, breast milk, especially the first secretion, which is called colostrum. Colostrum, I didn't test you on this, but it is a term that you should know. When you take OB, they'll go over colostrum. Colostrum provides a significant amount of immunity, and it's one reason why breastfeeding is promoted by mothers, because it confers a significant amount of immunities to the baby. These passive immunities last only for a few weeks or a few months. Then eventually the newborn, as their immune system develops, will begin to form their own antibodies. But in the interim, for that first, while in utero, and in the first few weeks, months after the baby's been born, the mother will transport or transfer the immunity in what's referred to as naturally acquired passive immunity. Now, some other terminology you should be somewhat familiar with. Serology, study of reactions between antibodies and antigens. In a clinical setting, you may hear nurse practitioner, physician may order serology on a patient to look at antibody levels, titers, for example. If you get the hepatitis B vaccine or the hepatitis A vaccine, one of the tests that you would have taken after you've done your series of vaccines is what's referred to as serology, or a titer level, to see if your body has formed the antibodies against hepatitis B. And so a physician, nurse practitioner would order serology. Another study, anti-serum, generic term of serum because it contains antibodies. So when we talk about anti-serum, anti referring to antibodies, so anti-serum relates to antibodies which would be contained in the serum. Globulins are basically proteins, types of proteins that's in the serum, and gamma globulin, you may hear that term used before. This is a serum fraction that contains antibodies. Now, gamma globulin is used as a treatment at times. Some individuals, for example, you get exposed to hepatitis B, and you're not vaccinated for hepatitis B. Now, it's too late to get the vaccine. You've been exposed. You got exposed today. Too late to get the vaccine, because if you get the vaccine, it takes several weeks for your body, several weeks, several months, for your body's immune system to develop the antibodies. So in the interim, what we can do is give you a protein-rich solution that contains antibodies, and hopefully that will help fight off the infection. Doesn't mean that you won't get the infection, but at least you can reduce the significance of the infection, the intensity of the infection, by administering gamma globulin. And that's commonly used in medical practices as well, being able to introduce or produce those. One of the ways to identify serum proteins, just as an aside, there is a method by which you can measure the weight of proteins. Each of us, as we develop antibodies in our system, the antibodies are basically proteins, and you can measure these levels and identify the particular proteins by doing what's referred to as an electrophoresis, which will basically weight out the charges of the certain proteins. You know, proteins typically have a charge, and you can measure the weight of the proteins to determine whether or not there are gamma globulins, beta globulins, and alpha globulins that are in the system. That's an aside. Back to the real meat of what we're talking about. In the immune response, should be familiar with these terms: acquired immunity, humoral immunity, cell-mediated immunity. Acquired immunity developed during an individual's lifetime. Humoral immunity involves antibodies produced by our B cells, and I'll talk about that in a little bit. Then cell-mediated immunity involves T cells, and we'll talk about T cells in just a little bit as well. Artificially acquired immunity. Acquired active immunity results from a vaccination. Now, you should know this. Artificial acquired immunity, given a vaccination, and you have developed then artificially acquired immunity. Makes sense. You know, the synthetically produced vaccine is artificial, and thus it will help you to develop antibodies. Immunization with a vaccine composed of an inactivated bacterial toxin. That's where the name toxoid comes from. Killed microorganism, that's another way. Living but attenuated or weakened microorganism can be injected as well. Now, understand this concept. If you get a vaccine, because you're going to be in a healthcare profession, all your family's going to ask you these questions. If I get the vaccine, am I getting the virus? Am I getting the bacteria? What is the vaccine doing? Well, typically, take a virus, for example, or take a bacteria, whatever. All we need is just the coating of that organism, because our body makes the immune system, our immune system rather, makes antibodies based upon the covering. The covering, remember when we talked about the glycocalyx, recognition proteins, recognition carbohydrates? You remember that, right? Okay. All right. That's what's recognized. So our immune system recognizes the covering of this microorganism as being foreign. Our immune system knows what our own glycocalyx is. It knows what our own cells are coated with. So our immune system won't typically attack our own cells. But when a foreign invader comes into our system, our recognition system recognizes that the glycocalyx, the protein covering, the protein covering of this foreign invader is different, and so then it will develop specific antibodies to attack that covering of that microorganism. And that's how our antibodies are formed. Now, in some cases, you may have heard of autoimmune type of conditions. Our immune system fails a little bit. It doesn't recognize our own cells as being from our own body, and will begin to attack our own cells. That's an autoimmune disease. That's not a good thing. But in this particular case, when we talk about artificially acquired immunities, basically all we need is a coating, the coat of that microorganism. If you can synthesize that in such a way and inject it in the body, doesn't mean you have to get the active virus or a bacterial infection. All you need is a piece of that organism, and our body's immune system recognizes that. And then if you ever are exposed to the virus or ever are exposed to that bacteria, our immune system recognizes it immediately as being foreign. We already have the antibodies in our system. The immune system revs up and produces a ton of antibodies, and boom, it's knocked out of our system. Now, some viral vaccines are produced by growing a virus that's weakened. You don't give it enough food source. You don't give it enough nutrients. So it grows the virus, but the virus is so weak that it can't cause disease. We use that as a vaccine as well. We inject this weakened virus into the system. Our immune system, again, recognizes it as foreign, produces antibodies, and will attack it. Or we can kill the microorganism, sterilize it, and inject it, and you can still have a vaccine. So typically, either we just give a little part of the microorganism's coating, or we can give a killed microorganism, or we can give a weakened microorganism, all of which is a form of immunization that will produce an artificially acquired type of immunity by our body. Artificially acquired passive immunity involves the injection of antibodies formed in a serum. That's a fluid that when the blood has been clotted, the blood cells and other matter has been removed from it, and then other people or from other animals. Most antibodies remain in the serum. The term is referred to as an antiserum, or means derived from fluid containing antibodies. Now you see this a lot on movies where somebody gets exposed to this wicked infection, but they don't get sick from it. And so the mad scientist is usually trying to find that person who got exposed to this bad disease and didn't get sick from it, because if you can get their serum, you can get the antibodies and find out why they didn't get sick and inject the anti-serum into other people and thus confer an artificially acquired passive immunity. The immunity was developed in somebody else or in another animal, another human, or an animal, it can be either one. But anyway, you want to get that antibody out of that serum and then give that to individuals so they don't get sick. Sometimes it's important because you can't figure out how some people get sick and how other people don't get sick. A lot of research is being done that in HIV disease now, because they are finding more individuals are getting exposed to the HIV virus, are developing antibodies to the HIV virus, and they're not getting sick. And they're not exactly sure why that is in some individuals versus others. They haven't made a lot of headway in that, and we certainly haven't produced an anti-serum, so to speak, yet. But certainly the potential is there. So this is artificially acquired passive immunity. Again, antibodies formed in the serum in somebody else or another animal. You remove the blood constituents of it, you just get the pure antibody, and then you're able to use that in a serum. Again, just as a review, serology, the study of antibodies, antigen, antibody-rich serum, referred to as gamma globulin, and then are the immune serum globulin. Then half-life of such of these antibodies is very short-lived. You'll see, again, in clinical settings that individuals, primary disease that I talk about is hepatitis B. People get exposed to it, and again, they did not get a vaccine, and they come down with fulminant disease. One of the ways you can try to treat them is to administer gamma globulin into their system. But again, you have to do it over a repeated period of time. Person may come in on an outpatient basis, receive one course of gamma globulin, then come back in 2 to 3 weeks, receive another course of gamma globulin, and what you're trying to do is just try to reduce the quantity and full amount of the disease. Now, acquired immunity, again, naturally acquired immunity resulting from an injection. Naturally acquired passive immunity, placental, transplacental, or via colostrum, which is basically the antibodies from the mom's milk. The first few weeks of milk production by the mom will transmit some immunities to the infant in a naturally acquired passive immunity. Artificial acquired immunity is an injection of an antigen, vaccination, then artificially acquired passive immunity, injection by antibodies. If you know this slide, then you should be able to answer the majority of the questions related to this on the exam. Humoral immune system involves antibodies that are dissolved in various bodily fluids, humors, the serum humors. This system responds to B cells. B cells are specialized lymphocytes that are exposed to particular antigens. Now, humoral immunity defends mostly against bacteria, such as bacterial toxins, viruses that are circulating in the body's fluid. So these humoral immunities will defend us against those types of an attack. Cell-mediated immune system involves specialized lymphocytes, such as T cells. T cells do not secrete antibodies, but they have antigen receptors that are attached to their surfaces. The cell-mediated immune system is the most effective against bacteria and viruses located within the phagocytic or the host cell. Also works against fungi, protozoas, and helminths. It also responds to cells or tissues as it perceives as being foreign or transplanted tissue and or cancer cells. We see this same type of a response take place. The nature of antigens and antibodies, the relationship here is the foreign invader, the antigen. Most antigens, some are called immunogens. We refer to them typically as antigens. Basically are proteins, lipoproteins, glycoproteins, or polysaccharides. I think that's a test question. If not, it was in the test bank, and I either used it or didn't. I can't remember. But when we do the review, I'll see if it's on there. But most antigens are proteins, lipoproteins, glycoproteins, or large polysaccharides. They are, for the most part, microorganisms or antigens such as pollen, egg white, blood cells, transplanted cells, tissue, and our organs. I bring up transplanted cell tissues because you have to recognize that the protein the protein, lipid, or carbohydrate components of the tissue that's being transplanted to an individual. You receive a heart transplant, or a heart-lung transplant, or a liver transplant, or a kidney transplant. The glycocalyx in the tissue, in the cells that you received, is different from the tissue or your own tissue. And so the recognition factors affect our immune system. Our immune system doesn't recognize that tissue as being the same and will launch an immune attack or an immune response against that particular tissue. And so when I say transplanted tissue, again, just as pollen is a foreign invader, pollen in the environment, we inhale that in, it gets into our system, and our immune system responds to it because it recognizes it as foreign. Egg whites in some people has a lot of protein, and some people can't tolerate that. And again, blood cells, you receive blood transfusions. Again, blood transfusions are donated blood, and though it's matched up very closely, same blood type as you have, but remember, it's still going to be a little bit different. And because it's a little bit different, our immune system may respond to the protein, lipoproteins, glycoprotein coding. The term epitopes, antibodies recognize and interact with antigenic determinants or epitopes on the antigen rather than the entire antigen. So there are just recognition factors, little pieces of this particular type of foreign invader that's recognized by our immune system. A bacterium or virus may have numerous antigenic determinants. These are the factors that make our immune system respond. Haptens, low molecular weight antigen, not antigenic unless it's first attached to a carrier molecule. Once the antibody against the hapten is formed, the hapten will react independently of its carrier. So if our system does recognize it, it will react. Penicillin is a good example of a hapten. Now, antibodies recognize and react to antigenic determinants on the epitopes. So this is just an illustration, again, taken from your book to illustrate some of the antigenic determinants where the antibodies will be able to recognize. Antibodies looks at these locations, quickly identifies this as foreign, and thus the antibody will attach. And this just illustration, it demonstrates to you the hapten. Again, that's taken out of your textbook. Now, antibodies are basically proteins. They're called immunoglobulins, Igs. Globulins are the protein that have a certain solubility. Characteristics are common in animal tissue. They are highly specific and will react typically to only one type of antigenic determinant. Now, that statement may, for the purposes of this course, that's a true statement. Sometimes when you're talking to a microbiologist, they debate that. I guess in the real sense that's not necessarily true. But for the purpose of this class, just remember these antigenic determinants are highly specific. So typically the antibiotic will only react to one antigenic determinant. Each antibody at least has two antigen binding sites, and then the valence is basically the number of sites on the antibody that's available for binding. A typical type of antibody has 4 proteins, has what we refer to as a light chain and 2 identical heavy chains. I'm not going to go into a lot of detail about this. I'm going to show you the structure of an antibody, but This is important when we're trying to identify some different types of immune diseases. Because there are identical chains, sometimes they have to measure through special testing whether this is a light chain or a heavy chain type of protein. At the end of the arms of the shape of the molecule, there is a Y-shaped molecule and then a variable region, which is a V region. The structure accounts for the ability of the antibodies to recognize and to bind to different antigens at these antigen binding sites. The stem of the antibody, or the long arm that comes down, the lower part of the Y arm is called the constant region or the C region. The stem of the Y-shaped is referred to as the FC stem region. Antibody molecules then can attach to a host cell by the FC region, and then complements, which are other agents that are in the bloodstream that help in the immune response, can also bind to this region as well. This is just an illustration out of your book. Again, I'm not going to spend a lot of detail on it, and I didn't ask you any questions about heavy chain or light chain, but basically this just shows you the structure of an antibody, and this just helps, these areas help to attach to the antigen or the foreign invader of our body. And this was what I was referring to as the V chain, heavy chain, V chain, the light chain, and then the stem region of the antibody. Again, important because it allows the antibody to find the site on the antigen to be able to bind, and you'll form an antigen binding site. Now, IG antibodies account for 80% of all antibodies in the serum. So this is the most common type, that's a test question, most common antibody found in our system, IG antibodies. 80% of all antibodies in the serum. Their monomers readily cross the blood vessel walls and enter into the tissue. Now, is that important? Well, yes, it is, because bacteria crosses into blood vessels. Viruses cross not only into blood vessels, but into blood cells. And so it's very important that the antibodies can get to where these foreign invaders are located at. Maternal IG crosses the placenta and can confer immunity to the fetus. So IG antibodies, the most common antibody, the most prevalent one that we see in our system. IG antibodies protect against circulating bacteria, viruses, neutralize bacterial toxins. Key point, neutralize bacterial toxins trigger the complement system. We're going to talk a lot about the complement system because it helps in this immune response, and then it binds to antigens, and it enhances the action of phagocytic cells. IgG is so long-lived that its presence may indicate immunity against a disease condition in the more distant past. So IG antibodies is important. Now, if you remember, I focused just a little bit on the IG ability to affect the bacterial toxins, neutralizing toxins. Remember, the toxin is a little bit different from the bacteria itself. Now, we talked about antibodies recognizing the covering of the bacteria or the covering of the virus. The toxin is different. The toxin is secreted by the organism once it's been infected. And the toxin can travel through the blood system and affect different areas of the body. Whereas the bacteria, for example, you eat a bacteria, the bacteria is in your gastrointestinal system, you treat it with antibodies, or there is especially like in traveler's diarrhea, we try to give not only a cathartic or a medicine that flushes out your system to get the bacteria out of your system, but we also may give activated charcoal that would absorb some of the toxin. Because the bacteria, even though you kill the bacteria, the toxin is still in your bloodstream. So an IgG antibody, not only will it attack the organism itself, the bacteria, but it also can go and neutralize the toxin. And so that's important because a lot of antibiotics that we give, oral antibiotics especially, will kill the bacteria, but it won't affect the toxin at all, and so the toxin can still cause a problem. So it's important to recognize that our own immune system, antibodies produced by our own immune system, is always the best way to treat, cure an infection, is through our own immune system, even though artificially we help it now by using artificially produced antibiotics. It's still our own immune system that works most efficiently. Just as a review, IgG antibodies, 80% of serum antibodies, Again, helps stimulate the complement system. Complement system complements the immune system, so it helps out. We see it in blood, we see it in lymph, we see it in intestines, we see it can cross the placenta. And I make a distinction there. In AMP1, we talked about the blood placental barrier, the blood brain barrier. Well, this is a barrier that typically antibodies can't get through. When you get meningitis, for example, one of the reasons why meningitis is so difficult to treat, artificially produced antibodies have difficulty crossing that barrier, the membrane barrier between the bloodstream and the cerebral spinal fluid. And so if you can't get the antibiotics, keep switching terms, if you can't get the antibiotics into the cerebral spinal fluid, you can't treat meningitis. And so typically you got to give very high levels of antibiotics, and you got to give so you got to get a little bit to get through that blood-brain barrier. So it's important to recognize that that's a barrier, and our own antibodies can cross that barrier, can cross that placental barrier, and thus enhance the ability of a person to get over infection. Phagocytosis is important. It enhances that process, neutralizes toxins and viruses, and also protects the fetus and the newborn, half-life of about 23 days. Now, IgM antibodies constitute about 5 to 10% of antibodies in the serum. IgM has a pentamer structure, as we talked about the Y chain and the long arm of the stem. The J is the joining portion of the antibodies. Again, I didn't focus on that a lot, but just remember, they constitute a small amount of the antibodies that we see in the system. IgM antibodies are the first to appear in response to antigens, but their concentration declines rapidly. IgM antibodies generally do not enter the surrounding tissue. IgM molecules are especially effective in cross-linking particular antigens, causing aggregation, causing them to clump together. It is the predominant antibody. It is the predominant antibody in the ABO blood group antigen reaction. That's, I believe, is a test question. Its presence in high concentration in patient makes it likely that the antibodies are associated with disease pathogen. So again, IgM antibodies, predominant antibody when we see the ABO blood group antigen reaction. Again, our immune system would recognize the coding of the red blood cells. If it doesn't recognize the coding of the red blood cells, our immune system will then attack those particular systems, or those particular cells. If it has a recognition factor, if it has the ABO compatibility, the histocompatibility, then you won't see our immune system respond. So again, that's why it's so important when you're receiving blood that they do what we refer to as a cross-match. Make sure you have the same type of blood that you're receiving is the same type of blood that you are, or the antigenic determinants have to be identical, otherwise our immune system is going to respond. Again, as a review, it's a pentamer. 5 to 10% of the serum antibodies, small as you can see in comparison to IgG. In the blood, lymph, B cells, they agglutinate microbes. In other words, they make microbes clump together. Antibodies produced in response to infection. Very short-lived, though. IgM antibodies only about half-life of about 5 days. Again, different from what we saw with IgG, you can have that lifelong. IgA antibodies account for 10 to 15% of the antibodies in the serum. IgA circulates in the serum as a monomer. Again, the structure similar to what we've discussed previously with the other antibodies, found on the mucosal surfaces of the body secretions, and as in colostrum, as we've already talked about. I think that's a test question. IgA found in mucosal surfaces in the body secretion. So you see that in the tear ducts, in sweat, in the mucus lining of the mouth, in the mucociliary layer of the lungs. IgA antibody found on mucosal surfaces. A secretory component protects IgA from enzymes and helps enter secretory tissue. The main function of IgA is preventing the attachment of viruses and certain bacteria to the surfaces of tissue. Again, dimer, 10 to 15% in antibodies. It's in secretions, mucosal protection, half-life of about 6 days. IgD antibodies, only about 0.2% of total serum. They are monomers found in the blood and in the lymph and in B cell. IgD functions in the serum. Little known about it, IgD antibodies on the surface of B cells act as an antigen receptor. Just a quick overview of the same. Now, IgE antibodies are also monomers, slightly larger than IgG, account for even a smaller percentage in the serum, 0.002%, bind to the Fc stem, the lower portion site, to mast cells and basophils. Now, why is this important? Well, the IgE response will bind to the mast cells, and we'll see how important the mast cells are in the inflammatory response. When an antigen reacts with IgE antibodies, the mast cells or the basophils release histamines and other chemical mediators in the allergic reaction. Important that you remember the relationship between IgE and this inflammatory response involving other cells such as the mast cells and the basophils. I'm almost sure that's a test question, because these chemical mediators in this inflammatory reaction dictate the inflammatory response. Now, we need an inflammatory response. We need this was what was discussed. I think Dr. Sloan went over this with you. This is what happens in an inflammatory response. Blood vessels dilatate. When the blood vessels dilate, it allows the cells that come in to fight infection to come into that area. Tissue gets red, tissue gets warm, but that's inflammation. That's not necessarily an infection. Doesn't mean that you're running a fever. In fact, most times you don't have to run a fever for inflammation. But it's a localized reaction where the body's immune system is trying to keep that inflammation, to try to keep that antigen in one spot. It doesn't want it to get to the rest of the system. It doesn't want any toxins to be released from the bacteria. It wants to localize that invading microorganism. So you get this inflammatory response. Tissue gets red. Blood vessels first dilate. The tissue gets red. The surface may get warm. No fever. Localized reaction. And it's the IgE antibodies that get triggered that release the histamines. Now, why is this? That's a good thing. Now, the bad part of the IgE and the mast cell response is if it's too intense. So you get a bee sting, and there's a toxin that's been secreted. The toxin is recognized as foreign. And then IgE responds. But too much IgE and then too many mast cells respond. What happens? You get too much vasodilatation, affects primarily the lungs, the mucous airway, and you get secretion of a tremendous amount of fluid. You get over-dilatation of blood vessels, which begin to block up the respiratory passageways. You have these huge responses throughout the body. You develop urticaria, big, large red whelps. Your throat swells up, your lungs swell up, and you can't breathe. So that's an anaphylactic type of reaction mediated by IgE and mast cells, but the reaction, even though it's a protective reaction, can sometimes be a bit too aggressive and cause us to have major problems. These inflammatory reactions can be protective, attracting IgG and phagocytic cells as well. Again, it's a monomer. Again, stimulates mast cells, basophils, allergic reactions. The other place I should have mentioned, I think that's a test question as well, and we'll talk about helminths when we talk about the digestive diseases, but parasitic infections by worms, by helminths, typically the response in our system, because helminths are live organisms that are inside our system, so the mechanism of our immune system is a little bit differently. You don't see IgG antibodies predominating because you can't form an antibody to a worm. But so we get a response by the IgE system, so you see eosinophils are elevated because the allergic reaction comes from the IgE antibodies. We get mast cells that will respond, but again, you can't form lifelong antibodies to worms. I mean, if you eat worms, you're going to get worm infection, and if you eat worms again, you can't form antibodies to it, and so our own body wouldn't be able to fight it off. You'd have to give something artificially. So anyway, B cells and humoral immunity. B cells, T cells, and macrophages all develop from stem cells. You all know what stem cells are. These are the immature cells in the bone marrow, sometimes in the skin as well. or deep in the integumentary system, I guess I should say, that can differentiate into whatever cell is needed for that time or for that crisis. Stem cells located in the bone marrow of adults or in the liver cells of embryos. Each B cell will produce antibodies against one specific antigen. Now, the term apoptosis, you're all familiar with as well. This is programmed cell death. Cells that die naturally are dismantled, and the remnants are disposed by phagocytosis. Cells that meet a violent end, that sounds pretty cruel, but a violent end called a necrotic death. Basically, blood supply or nutrient supply to a particular area of the tissue is cut off, and so that cell will die, and die a violent death. They spasm, they seize, and they burst open. So it's an ugly thing if you ever have to experience it. But basically, they will burst, and this bursting will cause an inflammatory reaction. The nature accomplishes apoptosis by a family of enzymes. The enzymes are called capases. I didn't ask you about that. But apoptosis, programmed cell death, is important to know. When cells die on their own or they die because they didn't get enough nutrients, then phagocytosis really gets rid of the remainder. IgM and IgD antibodies on the surface of B cells act as specific antigen receptors. So on the B cells, you have antibodies, IgM, IgD. Now remember, again, these aren't the predominant antibodies in the system, that's IgD. But they sit on the B cells, and they are specific antigenic receptors. So they look for the antigens. They're like the surveillance. They're sitting around, they're floating around the system, and they're just looking for an antigen somewhere so they can pounce on it and carry out their effect. Upon binding to an antigen, B cell proliferates and progresses, called clonal deletion. The immune system therefore shows what's referred to as a self-tolerance, meaning that it can distinguish itself from another organism. So self and or non-self are very important in being able to recognize these substances. So the immune system, again, is self-tolerant. You should be familiar with that term. An individual can respond to as many as 100 million or more different antigens. Mechanism that accounts for this is analogous to a generation of a huge number of words with a limited alphabet. So you have to be able to be versatile, our immune system does rather, and be able to recognize certain microorganisms and to be able to attack it. Clonal selection. Bone marrow gives rise to B cells. Mature B cells migrate to lymphoid organs. A mature B cell will recognize the epitopes, as we talked about earlier. And I didn't really go into any detail about clonal selection. You do need to know the concept of self-tolerance, though, because our immune system has to be able to recognize our own tissue as self and be able to recognize other invaders as non-self. So the process of destroying B and T cells that react to